Welcome to the Deep Dive, where we take the most complex research articles and scientific profiles and really distill the knowledge you need to be well informed.
And today, we are wrestling with a topic that, well, it just sounds like something straight out of science fiction.
It really does. We're talking about the ability to precisely edit the foundational code of life. I mean, rewriting the very blueprints of existence in any organism on Earth.
It's a monumental shift.
Yeah.
For decades, science was all about reading the genome. Now, we have the ability to go in and write new sections, correct errors, even delete entire passages.
And the figure right at the center of this revolution is Jennifer Doudna. Her journey is just as fascinating as the technology she helped create.
Absolutely.
So our deep dive today is built around a really comprehensive look at Doudna's career. And we're not just focusing on the 2020 Nobel Prize she won for CRISPR-Cas9 genome editing.
No, we have to go back further.
We're going all the way back to her origins. This inquisitive kid in Hawaii, the structural biologist obsessed with complex RNA shapes, to really understand the often overlooked foundational work that made all of this possible.
Our mission for you, the listener, is really to connect the dots. You need to understand the molecular mechanics of how this technology works, the high-stakes legal battles over its ownership, and the ambitious ethical framework Doudna is building to ensure this powerful tool is used responsibly.
We're tracing a whole scientific lifetime, from the lab bench to the Nobel stage and way beyond. Our goal is to make sure you walk away understanding not just the what of CRISPR, but the deeper how and why it matters for all of us.
So let's get started.
Let's unpack this journey, starting with her earliest inspirations.
It's so important to start with the environment that shaped her curiosity. Doudna was born in Washington, D.C., but her formative years, they really began when her family moved to Hilo, Hawaii. She was 7 years old.
And Hilo is such a specific, vibrant place. You can't live there without being just immersed in this unique, lush biodiversity.
Exactly.
The source material really highlights that this upbringing fostered a deep intrinsic fascination with the local flora and fauna. I mean, it's hard to imagine a better incubator for someone who would dedicate her life to understanding the mechanisms of biological systems.
And her parents were a huge intellectual influence. Her father had a PhD in English literature, and her mother, who taught history, held 2 master's degrees.
So the home environment was clearly one that valued learning and, intellectual pursuit.
But the real spark, the defining moment, they came in the 6th grade.
Right. This is the moment that's almost legendary in scientific biographies now. It's when her father handed her a copy of James Watson's 1968 book, The Double Helix.
A powerful gift. I mean, that book, which recounts the thrilling, sometimes messy discovery of the structure of DNA, it was a profound inspiration for her.
It introduced her to the idea that life itself could be understood through molecular structure.
Yeah.
That there was a blueprint.
And it's incredible to think of a 6th grader being captivated by that, by the molecular structure of existence.
And that seed of curiosity was then nurtured by some really excellent mentorship. Her 10th grade chemistry teacher at Hilo High School, a woman named Jeanette Wong, is cited as a major influence.
Right. She transformed that nascent interest into a really focused curiosity.
We can't overlook the power of those early teachers. There was also an encouraging lecture on cancer cells that sort of reinforced her career path.
But even with all of this early momentum, her confidence wavered when she got to Pomona College to study biochemistry.
A completely relatable moment of doubt, right?
Totally.
Our sources reveal that during her freshman year, Doudna actually considered dropping science entirely. She wanted to switch her major to French.
She felt overwhelmed by the difficulty of the general chemistry classes.
But here's the twist. It was her French teacher who encouraged her to stick with science.
It's a remarkable little historical footnote, isn't it? A single conversation that may have prevented a Nobel Prize-winning scientist from changing fields entirely.
So she overcame that self-doubt and graduated with her Bachelor of Arts in Biochemistry in 1985.
From there, the specialization really begins. She chose Harvard Medical School for her doctoral studies, completing her PhD in biological chemistry and molecular pharmacology in 1989.
And she was working under the renowned Jack Szostak.
Yes. And this is where her focus on nucleic acids truly locks in, but in a very, very specific way. Her dissertation wasn't just about DNA or RNA as, information storage. It was focused on increasing the efficiency of a self-replicating catalytic RNA.
That distinction, catalytic RNA, is so key to understanding her entire career trajectory.
It's everything.
For our listeners who might be familiar with basic biology, we typically think of RNA as a messenger, right? It carries instructions from DNA to the protein-making machinery.
And we think of proteins as the workers, the
Yeah.
enzymes, that perform all the actual labor like catalyzing chemical reactions.
Exactly. But Doudna was focused on ribozymes.
Right. RNA molecules that act as enzymes themselves, performing catalytic work. Her early dedication was to understanding RNA not just as a passive instruction manual, but as an active 3D functional machine.
So this intellectual foundation is really the unheralded precursor to the CRISPR breakthrough years later.
It absolutely is. She was already treating RNA as a potential tool long before Cas9 ever came along.
That sets us up perfectly for the next phase of her career. She knew RNA could perform this complex work, but she needed to figure out how. How was it structured to do that work?
So when Doudna began her postdoctoral work, the fundamental challenge facing her, and really the entire molecular biology community, was visualization.
Right.
You know, these complex RNA enzymes, these ribozymes, and scientists could measure their catalytic activity, see what they did, but they couldn't see their internal mechanics.
She realized she couldn't fully understand the function without actually seeing their three-dimensional structure. It's like trying to understand how a watch works just by listening to it tick.
That's a great analogy.
Yeah.
You eventually have to open the case.
And this challenge led her to the lab of Thomas Scheck at the University of Colorado Boulder starting in 1991.
And Scheck himself was a Nobel laureate for identifying catalytic RNA in the first place, so she was going to the source.
And her goal was monumental, to crystallize and determine the 3D structure of a ribozyme for the first time.
So why was solving the structure of a ribozyme so crucial? Why spend years on this painstaking, difficult work?
It was crucial to answer a really deep biological question. How similar or different are RNA-based catalysts to protein-based catalysts?
Okay.
I mean, if RNA performed the same duties as proteins, scientists needed to see how it folded and how it achieved chemical specificity. We needed to know if nature had evolved completely distinct structural rules for the two types of biological workers.
And the technique you have to use to see these molecules is notoriously difficult, right? We're talking about X-ray crystallography.
Can you just give us a conceptual idea of how that process works?
I can try. X-ray crystallography is essentially molecular photography, but instead of using visible light, you use X-rays. And first, you have to coax the molecule, in this case, a complex, floppy RNA strand, to arrange itself into a highly ordered three-dimensional crystal.
Which sounds impossible.
It's incredibly difficult. That process alone can take months or even years, especially for RNA, because it's often much more flexible than the rigid, well-behaved protein molecules scientists were used to working with.
So once you have a crystal, if you can get one.
If you get one, you bombard it with high-powered X-rays. As the X-rays pass through, they diffract or scatter off the atoms in the crystal. You capture the resulting pattern of dots on a detector, and then you use complex mathematical algorithms to reverse engineer the exact location of every single atom in that molecule.
So it's a process that requires massive computational power and, critically, extremely bright X-ray beams.
Yes, which is why the move she made later in her career was so strategic. But the initial breakthrough, that happened at Yale. She established her first independent lab there, running from 1994 to 1996.
This is the quiet foundational moment, right? The work that doesn't get the headlines but earned her scientific credentials.
Precisely. Her team succeeded in solving the 3D structure of the catalytic core of the tetrahemina group ribozyme.
This structure was the first complete blueprint of a major RNA enzyme. And this is where we need to dive into the specific molecular nugget that, fundamentally changed the field. What did they actually find inside this complex RNA fold?
What they found was a highly ordered specific cluster of 5 magnesium ions, and they were tucked deep inside the core of the molecule.
Magnesium ions.
Yeah, these magnesium ions were located within a specific domain of the ribozyme structure, and they were indispensable for the overall folding and stability of the molecule.
Wait a minute. We always hear about the hydrophobic core being crucial for protein folding. Proteins achieve stability by burying water-repelling amino acids in their center, isn't that right?
That's the traditional rule for proteins, yes. That's how they stabilize the complex shapes. But Doudna's team showed that this cluster of 5 magnesium ions was performing an analogous role for RNA.
How so?
Well, these positively charged metal ions organized the negatively charged phosphate backbone of the RNA, effectively allowing the molecule to fold into this complex, stable 3D shape.
So RNA uses ions in organic chemistry to create a core that functions structurally, like the organic hydrophobic cores of proteins.
It's a completely different chemical trick to achieve the same sophisticated architecture.
Wow.
It shows that RNA has its own intricate specific rules for self-assembly and catalysis. It's distinct from proteins, but equally capable of achieving complex functional folding. This wasn't just a pretty picture. It was a realization that RNA was a powerful, independent architect of life.
And this success immediately established her expertise in structural biology.
It did. It led to further work on other critical ribozymes, like the hepatitis delta virus ribozyme, and larger complexes, such as the signal recognition particle.
So her need for even higher resolution imagery and more intense X-ray beams prompted her move.
Exactly.
Yeah.
In 2002, she joined UC Berkeley, gaining access to the powerful synchrotron at Lawrence Berkeley National Laboratory. This was a critical step. A synchrotron is essentially a massive particle accelerator that creates X-rays bright enough to produce the high-resolution diffraction patterns needed to map these incredibly dense structures.
So by 2006, when the CRISPR story really starts, she isn't just a smart scientist. She's one of the world's foremost experts in understanding the 3D structure, folding, and function of complex nucleic acids.
She had the exact toolkit required for the next massive leap.
The transition from structural biology to genome editing, it started not with an epiphany in a darkroom or a crystallography lab, but with a surprisingly modern tool.
A Google search.
I still love that detail. In 2006, Gillian Banfield, a Berkeley colleague studying microbial genomics, was looking for someone who understood RNA interference, RNAi, to help interpret these mysterious repeating DNA sequences she kept finding in bacteria.
So she literally searched RNAi in UC Berkeley. And Doudna, because of her extensive work on RNA structure, came up right at the top of the list.
It just shows the sheer weight of Doudna's prior work. That initial contact led to Doudna investigating this unusual system, CRISPR.
Right. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which is a mouthful.
A total mouthful. At the time, it was just a genomic curiosity.
And it was known that the sequences acted as a kind of bacterial immune system. When a bacterium survived a viral attack, it would snip a piece of the viral DNA and store it in its own genome, right between those short repeating sequences.
It was like a molecular mugshot database.
That's the perfect way to describe it. The system itself was first noted by Yoshizumi Ishino way back in 1987, but Francisco Mojica later characterized it, suggesting it was an immune system. They basically discovered the lockbox.
But the key was missing.
The key was missing. And the breakthrough that unlocked the whole thing happened in collaboration with Emmanuelle Charpentier, a Swedish microbiologist Doudna met at a conference in 2011.
Their core realization, published in 2012, was that this microbial immunity system, which uses the Cas9 protein as a molecular scissor, could be simplified and repurposed.
They showed it was a universal, programmable editing tool.
Okay, let's break down the mechanics, because this is the core of the invention. In nature, the Cas9 protein works with two RNA molecules. There's the CARNA, the CRISPR RNA, which carries the targeting sequence matching the viral DNA.
And then there's the TRAC RNA, or transactivating CRISPR RNA, which acts as a kind of scaffold to activate the Cas9 protein.
So Doudna and Charpentier's genius was simplifying this complex two-part system into one single engineered molecule, the single guide RNA, or sgRNA.
Yes, this sgRNA contained both the targeting information and the scaffold necessary to recruit and activate the Cas9 enzyme, all in one piece.
That simplification was revolutionary. It means if you want to cut a piece of DNA, you just synthesize an sgRNA that matches your target sequence, pair it with the Cas9 protein, and put it in the cell. The guide RNA directs the Cas9 to the precise location in the genome.
And this brings us to one of the most crucial pieces of molecular detail that often gets overlooked, but is so essential for you to understand. And that's the protospacer adjacent motif, the PAM sequence.
The PAM sequence. Okay, why is that specific short sequence so important?
The PAM sequence, it's usually just 2 to 6 base pairs long, and it's the molecular handshake. It's a specific sequence that must exist right next to the target site in the host DNA.
And Cas9 won't cut unless it sees that.
Cas9 does not cut unless it first recognizes and binds to that PAM sequence. This acts as a critical safety feature for the bacteria. It ensures Cas9 only targets foreign invaders, and it prevents the system from slicing up the bacterium's own stored CRISPR sequences.
Okay, so Doudna and Charpentier didn't just show that Cas9 could cut. They showed that because the guide RNA was so easily swappable, the entire system could be aimed at any desired sequence in the genome of any organism.
As long as there was a PAM sequence nearby.
Right. It demonstrated full programmability.
Exactly. That flexibility is why it's called a revolution. You don't need years of re-engineering specific enzymes for specific targets anymore. You just change the RNA sequence in the guide molecule. This ease, precision, and speed transformed the landscape of biological research almost overnight.
The ensuing recognition was pretty much inevitable. They were jointly awarded the 2020 Nobel Prize in Chemistry for this development, solidifying its place as one of the most significant biological breakthroughs ever. A tool, this powerful one, that allows us to edit evolution itself. It naturally explodes into three distinct and very high-stakes theaters: immense applications, necessary ethical debates, and brutal intellectual property battles.
Yeah, that's a good way to put it. Let's start with the incredible potential.
The applications are immediate and profound. They touch everything from fundamental research to global food security. Beyond just understanding basic cell function, which has already accelerated so rapidly, we see immediate therapeutic promise.
Our sources list treatments for severe inherited conditions like sickle cell anemia, cystic fibrosis, Huntington's disease.
The precision allows scientists to correct the single faulty gene responsible for the condition. It's incredible.
And it's not just genetic disorders. I mean, consider HIV. The virus integrates its DNA into the host cell's genome. CRISPR offers the possibility of precisely excising that viral DNA from T cells, effectively curing a persistent infection that conventional drugs can only manage.
And then there's the massive applications in plant and animal research, dramatically shortening the time needed to develop sustainable, disease-resistant crops.
But the magnitude of this power demanded an immediate and careful ethical framework. And Doudna was one of the first scientists to really sound the alarm on the technology she helped create.
Her role as a thought leader here is just crucial. Recognizing how fast the field was moving, Doudna, along with other leading biologists, called for an immediate, worldwide moratorium on certain clinical applications of gene editing.
Very early on, she helped force the global community to pause and consider the implications.
The key ethical distinction here, which is essential for our listeners to grasp, is the difference between somatic and germline editing. How does Doudna draw that line?
She strongly supports somatic gene editing. Soma means body. These are changes made to non-reproductive body cells, like liver cells or blood cells.
Okay.
These edits are confined to the individual patient, they treat the disease, and they do not get passed down to the next generation. This is essentially just treating the individual with a highly advanced form of medicine.
But she opposes germline gene editing.
Yes, germline editing involves making changes to embryos, eggs, or sperm, the reproductive cells. Any edit made here is inheritable. It gets passed down to all future generations.
And this is where the scientific community's deepest ethical concerns lie.
It is. You're moving beyond therapy for an individual to potentially altering the human gene pool forever. Doudna has been really emphatic in her opposition to germline editing, especially for non-therapeutic enhancement purposes.
It shows a scientist fully aware that she has created a tool that, in her words, holds the unthinkable power to control evolution.
And that awareness is why she stepped into the ethical arena, understanding that the science was just moving faster than the societal consensus.
Now, in parallel with this ethical debate, the question of who owned this foundational technology, it became a legal cage match.
It really did. Yeah. The high-stakes intellectual property conflict that defined the mid-2010s. This was the complex patent war between the UC Berkeley team, led by Doudna and Charpentier, and the Broad Institute of MIT and Harvard, primarily featuring Feng Zhang.
The stakes were astronomical. We're talking potentially trillions of dollars in future royalties.
So what was the core difference in their patent claims?
The conflict revolved around the timeline and the context of application. The Doudna UC Berkeley team's claim was based on the fundamental description or conception of the universal programmable system.
So they showed how to engineer the sgRNA to cut any DNA in a test tube, and they detailed the theory of how it would work in more complex cells.
Right. And the Broad Institute's argument?
The Broad Institute, a few months later, demonstrated that the Cas9 system could successfully edit genes, specifically in eukaryotic cells, complex cells, like human cells, that contain a nucleus.
So their legal argument centered on reduction to practice.
Exactly. that they were the first to provide physical, published evidence that the system worked in the complex environment of human cells, which was an environment that had been considered much harder to penetrate.
So the argument boiled down to who should get the patent? The inventor who conceived of the fundamental tool and explained how it could be used universally.
Or the team that first provided empirical proof that it worked in the most economically valuable cells, namely human cells.
That's the core legal and philosophical divide. In 2017, the U.S. Patent Trial and Appeal Board initially favored the Broad Institute regarding the specific claims involving application in eukaryotic cells.
It was a complex ruling because it suggested that applying a known system to a new environment could be considered a non-obvious, patentable invention, separate from the original invention of the system itself.
But that wasn't the final word, was it?
No, far from it. The UC Berkeley group eventually received broader patents covering the general technique of programmable gene editing. And on top of that, the European landscape offered a completely different result.
What happened in Europe?
The European Patent Office disallowed the Broad Institute's initial claim due to a procedural flaw. It was related to who was listed on the patent application versus the original scientific publication.
A legal technicality.
A very important one. It shifted the momentum significantly, suggesting the UC Berkeley group would likely hold stronger patent positions in Europe for the general method. The whole patent landscape is still this multifaceted, high-value conflict.
Despite that intense rivalry, which Doudna described at one point as feeling like a divorce, this period fueled immediate commercial efforts.
Yeah, she jumped into entrepreneurship almost instantly to translate the technology into usable treatments and diagnostics. She recognized that foundational research, no matter how profound, needs a mechanism to reach patients.
She co-founded Caribou Biosciences in 2011, even before the seminal paper was published.
And she was briefly involved in Editas Medicine, which also focused on gene therapy.
Briefly. She was a co-founder in September 2013 alongside Zhang, but she resigned in June 2014, reflecting the complexity and strain of that early competitive environment.
But she shifted her focus, co-founding Intellia Therapeutics, which is explicitly focused on in vivo gene editing, delivering the CRISPR tools directly into the patient's body to treat genetic diseases.
And critically, she later co-founded Scribe Therapeutics. They're focused on next-generation CRISPR tools. Specifically, Scribe pioneered Cas9, a newly discovered family of Cas proteins.
Why are these next-generation Cas proteins so important? I mean, isn't Cas9 the gold standard?
Cas9 is a very large protein, which makes it challenging to package into delivery vehicles, like the viruses that are used to shuttle the gene editor into human cells.
Okay.
Cas9, which Scribe focuses on, is significantly more compact. Its smaller size means it can be delivered more easily and efficiently, opening up new avenues for treatment where size constraints were a major hurdle.
So she's not just focused on application, but on constantly improving the tool itself.
Right. Her entrepreneurial endeavors really demonstrate that the goal isn't just winning a Nobel Prize, it's ensuring the mechanism gets out into the world to solve real problems.
You know, for most scientists, the Nobel Prize is the culmination of a career. But for Doudna, it seems to have been more of a launching pad.
A launching pad for massive institution-building work focused on societal impact.
And her primary vehicle for this is the Innovative Genomics Institute, or IGI. She co-founded it in 2014 and serves as its director, leveraging this collaboration between UC Berkeley and UCSF.
The IGI's mandate is huge. It's designed to accelerate the application of genome editing technology to three pillars of major societal problems: human health, sustainable agriculture, and climate change.
That goes far beyond basic lab science.
It does. So what does that kind of ambitious research look like on a day-to-day level in her lab today?
Well, they maintain a focus on optimizing the tool. They continue to study the structure and function of diverse CRISPR-Cas systems, like Cas9 and Cas12, always looking for a better, more precise molecular cutter.
But the applied work is massive.
Oh, yeah. They're dedicating significant resources to developing novel and efficient delivery mechanisms, just figuring out how to safely and precisely target the molecular scissors to the right cells in the body.
And you mentioned a fascinating novel technique they're researching: precisely editing microbiomes. That seems incredibly complex.
It is, but the potential is enormous. Instead of editing human cells, they're developing ways to use CRISPR to precisely edit the complex microbial communities that inhabit our bodies or our soil.
So you could, eradicate antibiotic-resistant bacteria?
Potentially. Or you could engineer soil microbiomes to sequester carbon more effectively, directly addressing climate change and human health challenges at the same time.
That focus on immediate large-scale application was starkly illustrated by her response to the global COVID-19 crisis.
Absolutely. The pandemic hit in early 2020, and Doudna immediately mobilized the IGI infrastructure. She quickly organized an effort to repurpose CRISPR-based technologies to address the virus.
They didn't just study it, they acted as a public health service. The IGI testing center ended up processing a massive volume over 500,000 patient samples.
Right from the UC community and surrounding critical areas, including essential farm workers in the Salinas Valley.
It really proved the power of having such a dynamic research infrastructure ready to pivot. And on top of that, her diagnostics company, Mammoth Biosciences, played a key role.
Yes, in developing a rapid, CRISPR-based point-of-need diagnostic test for COVID-19.
This diagnostic test was noted as being much faster and less expensive than the traditional QRT-PCR tests that were being used widely at the time.
It showed that CRISPR isn't just about editing genes, it's also a highly sensitive biosensing tool.
And that's what Mammoth Biosciences focuses on.
Exactly. She co-founded it in 2017, and it focuses entirely on leveraging this sensing capability of CRISPR to create rapid, precise diagnostic tests across various fields: healthcare, agriculture, biodefense, environmental monitoring.
So it's a crucial commercial strategy, using the precision of Cas proteins for rapid detection, not just for cutting.
Right. And beyond her direct work, her influence spans the entire scientific and financial landscape. She holds major advisory roles, guiding investment decisions for colossal entities like Altos Labs, Johnson & Johnson, and Sixth Street Partners.
That kind of institutional weight shows that the decisions she makes ripple across global capital investment in biotech.
And here is a striking testament to her stature in computational science, even though she is fundamentally a biochemist. The announcement in 2025 that a new supercomputer was being named after her.
The Doudna supercomputer. Intended for the National Energy Research Scientific Computing Center at LBNL, it's going to be the successor to the Perlmutter supercomputer, focusing on integrating AI into massive scientific research calculations.
To have a major piece of national computational infrastructure named after you shows an influence that just transcends your original discipline. It acknowledges the computational revolution that CRISPR started.
And to ground this monumental figure, we can look at some of the personal details that humanize her ambition. She's married to Jamie Kate, who is also a distinguished professor at Berkeley.
And his own work intersects perfectly with the IGI's mission. He focuses on using gene-editing yeast for biofuel production. It's a family dedicated to applied science.
The source material even notes their move to Berkeley in 2002 was partially because Kate preferred the less formal, research-focused environment of the West Coast.
And, continuing the tradition of curiosity and technological focus, their son, born in 2002, is currently attending UC Berkeley.
Studying electrical engineering and computer science. It's a powerful legacy of foundational and applied science.
So we've navigated the full scope of Jennifer Doudna's career. The key takeaway for you, the listener, is really understanding that the greatest applied breakthroughs often spring from the most detailed, basic, and seemingly obscure fundamental research.
Right, the structural knowledge she gained, painstakingly solving the 3D architecture of ribozymes and realizing that RNA could fold into these complex functional shapes using elements like clustered magnesium ions. That was the crucial foundation.
It was everything. Without that deep expertise in nucleic acid structure, the breakthrough of engineering the single guide RNA to program Cas9 simply couldn't have happened.
Her journey really confirms that basic curiosity, fueled by books like The Double Helix, ultimately culminated in the creation of this universal tool.
A tool that addresses humanity's most persistent challenges, from inherited disease correction to developing climate-resilient agriculture.
And as she co-authored in her book, A Crack in Creation, the power she helped unleash truly is an unthinkable power. She remains focused, not so much on the science now, the science is largely solved, but on the sociological challenge of access and equity.
She has stated very clearly that she is, and I'm quoting here, concerned that the benefits of the technology might not reach those who need it most if we're not thoughtful and deliberate about how we develop the technology.
So the challenge has shifted from making the tool work to ensuring the tool benefits all of humanity, not just privileged segments.
Exactly.
So we leave you with this final, provocative question to consider. If CRISPR technology allows us to fundamentally solve issues in health, agriculture, and climate change simultaneously, and this technology is largely owned and commercialized by private companies, whose responsibility is it? the scientist who creates the tool, the government that regulates it, or the market that commercializes it, to actively ensure the benefits of this unthinkable power are distributed equitably across the globe.
Something to reflect upon as we witness the code of life continuing to be rewritten.
Thanks for diving deep with us. We'll see you next time.